Number 1, the Barreleye. The glass cockpit. Macropinna microstoma hovers at 600 to 800 meters in the mesopelagic zone. It is small, about 6 inches long, and its head is completely transparent. A fluid-filled dome covers the top of its skull like a pressurized cockpit, surviving roughly 80 times atmospheric pressure. Inside that dome, 2 large tubular eyes glow bright green. They point straight up. Marine biologist W. M. Chapman first described this species in 1939. But for 65 years, no one knew the dome existed. Every specimen hauled to the surface arrived with its head destroyed. The transparent shield is so fragile that it disintegrated during collection. It was not until 2004 that researchers Bruce Robeson and Kim Reisenbichler at the Monterey Bay Aquarium Research Institute captured live footage using a remotely operated vehicle off the coast of Central California. That footage changed everything. Here's what misled scientists for decades. The 2 small spots on the front of its face look like eyes. They are not. Those are olfactory nares, the fish equivalent of nostrils. The real eyes are the green orbs inside the dome, pointing upward. This false face fooled researchers into constructing an entirely wrong model of the animal's vision. The tubular eyes can rotate from a vertical scanning position to a horizontal targeting position. When the fish spots prey overhead, it pivots its eyes forward and swims upward. The green pigment in the lenses serves a specific filtering function. It blocks ambient sunlight trickling down from the surface. This allows the Barreleye to isolate bioluminescent signatures from prey animals drifting above. The technique is called silhouette hunting. The fish watches for organisms backlit against faint downwelling light. To avoid being hunted the same way, the Barreleye produces its own ventral light, counter-illumination camouflage. It erases its own shadow from below. Its mouth is tiny. That is not a design flaw. It is a specialization. The Barreleye picks small crustaceans directly off the stinging tentacles of siphonophores, those giant colonial jellyfish that trail driftnet-like tentacles through the deep. It is a siphonophore pirate. It steals food rather than hunting independently. Its large flat fins allow precise incremental movement without creating turbulence. A specialized gas bladder provides neutral buoyancy. The fish can hover motionless like a drone on station. Its metabolism is slow. Its digestive efficiency is high. It survives on minimal calories in what researchers call deep-sea food deserts. High-density optic nerves suggest extreme visual data processing speed. Its DNA profile reveals extreme specialization in genes responsible for retinal protein synthesis. The mainstream theory treats the Barreleye as a product of deep-sea selective pressures. Darkness, pressure, and scarce food drove radical cranial adaptation over millions of years. A second theory draws on comparative anatomy. Structural parallels with Cambrian organisms like Opabinia, which also had forward-projecting eyes and unusual head structures, suggest the dome may echo an ancient body plan, one that most lineages abandoned over 500 million years ago. A third, more speculative theory circulates in fringe biology. The glass-domed pilot aesthetic is cited as the strongest visual case for convergence with imagined extraterrestrial morphologies, not evidence of alien origin. But a reminder that when we imagine life on other worlds, we keep inventing something that already exists in our own ocean. Only one species occupies the genus Macropinna. Its lineage branched off millions of years ago. The cranial architecture it carries is found nowhere else on Earth. Whether that makes it an evolutionary dead end or a perfected design depends entirely on your framework, but what lives inside its transparent head is not the strangest intelligence in the deep sea. Number 2, the Mimic Octopus. The problem of intelligence. Thom octopus mimicus does not simply change color. It impersonates at least 15 different species, not random shapes, specific dangerous animals. When threatened by damselfish, it spreads its arms wide, shifts its color pattern, and mimics the venomous lionfish, complete with the appearance of radiating spines. When a different predator approaches, it tucks its arms back, flattens its body, and undulates across the seafloor to replicate a toxic soul. For sea snake mimicry, it hides 6 arms in its burrow and extends 2 in opposite directions with black and white banding. Costume switches happen in under 1 second. That is faster than any known terrestrial vertebrate color change. The skin contains millions of chromatophores, individual pigment cells that expand and contract on command. Papillae cells change the actual texture of the skin's surface. Muscular hydrostatic pressure creates horns, bumps, or smooth surfaces. It can look like coral. It can look like a rock. It can look like something that will kill you. Here's where it gets strange. The Mimic Octopus has a decentralized nervous system. Each of its 8 arms contains its own ganglia, functioning as a semi-autonomous mini-brain. The central brain coordinates, but the arms can operate independently. They taste what they touch. They make local decisions. The cognitive complexity this implies is staggering. Selecting which predator to impersonate based on the specific threat approaching requires something resembling threat assessment. Some researchers argue it suggests a rudimentary theory of mind. The animal appears to model what its predator expects to see. It lives on murky, featureless estuary floors in Indonesia. There's nothing to hide behind. Mimicry is the only viable defense. Its W-shaped pupils enable high-contrast vision in low-light conditions. Its RNA editing capability is extraordinary. It can recode its own RNA to adapt protein function to temperature changes without altering its DNA. That is adaptation happening at the molecular level, within a single generation. If an arm is lost, it regrows with full neurological function and mimicry capability intact. The metabolic cost of constant shapeshifting is immense, demanding high protein intake. Its inking mechanism releases chemical deterrents alongside standard ink. Encounters between mimic octopuses are rare and highly ritualized. The mainstream explanation treats this mimicry as an evolved, genetically hardwired response suite. Natural selection refined it over millions of years. Each successful impersonation increased survival rates, and the behavior became encoded. A comparative evolutionary theory frames cephalopod intelligence as a parallel track to vertebrate cognition, a second genesis of complex problem-solving that diverged over 500 million years ago. Octopuses and humans share a common ancestor, but it was a flatworm-like creature with almost no nervous system. Everything the octopus brain does, it invented independently. Then there is the fringe hypothesis. In 2018, a group of 33 scientists published a paper in Progress in Biophysics and Molecular Biology suggesting that octopus ancestors may have arrived on Earth via cometary material. The panspermia hypothesis applied to cephalopods. The paper was controversial. Most biologists rejected it. But the authors pointed to the octopus's alien-like intelligence, liquid morphology, and RNA editing as traits difficult to explain through conventional gradualism alone. Whether you find that plausible or absurd, the Mimic Octopus remains a cognitive puzzle wrapped in shape-shifting skin. Its intelligence and form are staples of science fiction alien design. That says something about either our imagination or our pattern recognition. But if you think an individual animal is hard to classify, wait until you encounter something that might not be an individual at all. Number 3, the Giant Siphonophore. One animal or a civilization, Priadubia, apolemia. These are not single animals, they are floating colonies of thousands of specialized individuals called zooids. A giant siphonophore can stretch up to 150 feet. That is over 45 meters. It may be the longest animal on Earth. Or it may not be an animal at all, depending on how you define the word. Every zooid in the colony is a genetic clone of the original founding organism, called a pro-bud. But they are not identical in function. There are hunting zooids armed with stinging nematocysts. There are digesting zooids that process captured prey and distribute nutrients. There are reproducing zooids called gonophores that handle egg and sperm release. And there are propulsion zooids called nectophores, jet propulsion bells that pulse in synchronized rhythm to move the entire chain through the water. A central stem connects them all. Nutrients flow through this canal like blood through a circulatory system. Nerve signals travel along it to coordinate the swimming bells into unified pulses. When hunting, the colony arranges itself into a spiral formation to maximize the surface area of its predatory web. Thousands of stinging cells create a massive electrified drift net capable of paralyzing hundreds of organisms simultaneously. Specialized zooids pulse with bioluminescent light to attract fish, neon signs in the dark. Most siphonophore species glow blue or green. Some produce rare red light. Since most deep-sea organisms cannot detect red wavelengths, red light producers effectively operate on a private channel. A pneumatophore, a gas-filled float, maintains vertical position. The entire organism is so fragile that it disintegrates on contact with human tools or collection nets. Its primary depth range is the midnight zone, 1,000 to 4,000 meters. If the chain breaks, fragments can survive as smaller, independent colonies. The lineage is ancient. Siphonophores represent one of the oldest evolutionary lines of complex life on Earth, largely unchanged for hundreds of millions of years. The mainstream theory treats colonial organisms as a natural extension of cellular cooperation, the same principle that built multicellular life from single-celled ancestors, just scaled up another level. A comparative theory looks deeper into the fossil record. Siphonophores may preserve body plan logic from the Ediacaran period, over 540 million years ago, offering a window into organizational strategies that predated the Cambrian explosion. The speculative interpretation goes further. The biological spaceship concept frames the Siphonophore as a self-sustaining modular megastructure, a floating civilization of clones, each performing a role, connected by a shared nervous system and nutrient network. It parallels theoretical designs for generation ships or self-replicating probes. The question of whether a Siphonophore is one organism or many has no settled answer. And that unresolved question connects to something even more unsettling. What happens when we encounter a body plan so alien we have almost no data at all? Number 4. The Bigfin Squid. The geometry of the unknown. Magna Pinna. The name means large fin. But the fins are not the disturbing part. The tentacles are. They extend from the body at precise 90-degree angles, creating elbow-like bends that look engineered rather than grown. Each tentacle stretches 15 to 20 times the length of the mantle. Total estimated length, including tentacles, exceeds 26 feet. Over 8 meters. This animal has been filmed at over 6,000 meters. The hadopelagic zone. That is deeper than most of the ocean floor. The lower portions of its tentacles are incredibly thin and sticky, unlike the clubbed tentacles of other squids. The large heart-shaped mantle fins provide gentle propulsion. The creature drifts rather than swims. In ROV footage, it appears to hover motionless with tentacles trailing below like impossibly thin wires. The feeding theory holds that the tentacles function as flypaper. They passively catch tiny particles of marine snow, the constant downward drift of organic debris that is the primary food source in the abyss. Only a few dozen sightings have been recorded across all deep-sea expeditions worldwide. The family Magnapinidae was not even recognized as distinct until the late 1990s. No large adult specimen has ever been captured. Everything known about adult bigfin squid comes from video. Skin pigmentation is pale pink or ghostly white. There's no need for surface-level camouflage in permanent darkness. Pease lights, specialized chemical stabilizers, reinforce cellular structures to prevent protein crushing under pressure. There's no functional ink sac. At total darkness depths, ink serves no purpose. Highly sensitive mechanoreceptors likely detect vibrations in water that is otherwise perfectly still. The mainstream theory interprets the elbow posture and filament tentacles as an ultra-low energy ambush feeding strategy adapted to near-zero food availability. A comparative theory suggests Magna Pinna may represent a cephalopod body plan that flourished in deeper Mesozoic oceans and has since been confined to abyssal refugia, pockets of stable environment that preserve ancient designs. The speculative reading focuses on what many observers note instinctively. It looks too fragile for Earth, too geometric, too still. The spindly horror trope, as some writers call it, an entity that appears designed for a lower gravity environment but thrives in the pressure equivalent of being buried under miles of rock. Cinematic comparisons are inevitable. The tripods from War of the Worlds, the sentinels from The Matrix. But those were imagined. This is real. And unlike the bigfin squid, which hides by being almost nothing, the next creature on this list hides by being almost invisible. Number 5. The Glass Octopus. Invisibility as biology. Vitroledinella richardi is nearly 100% translucent, a living fiber optic organism. You can see through its head, through its body, through its arms. Only 3 things remain opaque: the digestive gland, the eyes, and the optic nerve. These cannot be made transparent because of their chemical composition. Everything else is glass. The eyes are rectangular and tubular, specialized for extreme light sensitivity in the bath apelagic zone, 1,000 to 4,000 meters. Inside the transparent head, a yellow path is visible. That is the actual neural bundle connecting the eyes to the brain. You are looking at a living nervous system through a living window. The stealth advantage is total. Casting no shadow makes it invisible to predators hunting from below using silhouette detection. The gelatinous architecture is firm enough to maintain structural integrity under crushing pressure, but the organism relies entirely on surrounding water pressure to hold its shape. Remove it from the deep, and it collapses. It melts. Females carry eggs within the mantle cavity, brooding them until they hatch as glass larvae. The animal participates in the ocean's great vertical migration, moving up the water column at night to feed. It has evolved away from nearly all chromatophores. Unlike surface octopuses, it has abandoned color-changing cells entirely. Pisolites prevent protein folding failure at depth. The centralized brain is optimized for visual processing. A tiny radula, a serrated tongue, rasps flesh from small, deep-sea crustaceans. Internal organs are arranged with mathematical precision to minimize the visual profile. Jet propulsion via siphon, combined with delicate webbing undulation, provides locomotion. The mainstream theory holds that transparency evolved as the ultimate passive camouflage in open water where there is nothing to hide behind. The comparative reading frames this as a radical evolutionary departure. The glass octopus abandoned the entire cephalopod defensive toolkit. No ink, no chromatophores, no shapeshifting. A total commitment to a single strategy: invisibility. The speculative theory notes that through an ROV lens, the glass octopus resembles a hologram, a life form made of liquid glass. It prompts the question of whether transparency at this level suggests optimization for a medium other than seawater. If the glass octopus represents the most passive defense strategy in the deep sea, the next animal represents the most violent offense. Number 6. The Bobbit Worm. Ambush engineering. Eunice aphroditoise buries its entire body in seafloor sediment, up to 10 feet, 3 meters of segmented, iridescent worm hidden beneath the sand, with only its sensory crown exposed. 5 antennae detect chemical and vibrational shifts in the water above. It waits, sometimes for years. The strike is among the fastest in the animal kingdom. The jaws launch with enough velocity to snap a fish in half on impact. The compound jaw system features multi-part mandibles called maxillae that operate like a mechanical claw. The neuroreflex arc that triggers the strike runs through the peripheral nervous system. It executes faster than conscious vertebrate thought. The exoskeleton shimmers with iridescent rainbow metallic coloring. Hundreds of body segments each carry their own parapodia, limb-like appendages used for gripping the walls of the burrow. A venomous cocktail paralyzes prey on contact. The neurotoxin initiates external digestion before the food is fully consumed. Chitinous armor plating protects the worm from the struggles of captured animals. This creature can live for decades. It grows slowly, remaining underground, waiting for single high-calorie targets. Despite lacking complex eyes, its entire body detects light changes through photoreceptive skin. Bright red, branch-like gills on each segment extract oxygen from silty water. It is known to attack ROVs and metal probes with no fear response to larger objects. Reproductive swarming occurs during certain lunar cycles through synchronized gamete release. Prey skeletons are either ejected or pulled deep into the burrow. Its DNA shows extreme genetic divergence from other polychaete worms, suggesting a very ancient lineage. Mainstream theory classifies it as a highly specialized ambush predator refined by millions of years of benthic selection pressure. The comparative theory points to trace fossils. Structures resembling bobbit worm burrows have been found in Miocene-era sediment, suggesting this hunting strategy has persisted largely unchanged for at least 20 million years. In modern terms, that means this animal was already doing exactly what it does today while great apes were still evolving. The speculative reading draws on the obvious, the sandworm parallel. Its mechanics, longevity, and subterranean lifestyle mirror science fiction archetypes so precisely that some fringe thinkers ask whether fiction is unconsciously describing something real. But if the bobbit worm blurs the line between animal and machine, the next creature blurs the line between animal and plant. Number 7. The sea sheep. The animal that became a plant. Costacea kuroshimi is one of the only animals on Earth capable of true photosynthesis. It is a sea slug, rarely exceeding 5 millimeters to 1 centimeter in total length. It grazes on Vrainvillia algae and does something no animal should be able to do. It incorporates the algae's chloroplasts into its own tissues. This is kleptoplasty, chloroplast theft. Once stolen, those chloroplasts continue to function. The sea sheep can survive up to 2 months on sunlight alone without eating. The serrata, leaf-like organs on its back, house the stolen chloroplasts. Chlorophyll concentration in translucent tissues gives the animal a green glow. It pulls carbon dioxide from the water and converts it to sugar. It is a tiny carbon sink. An animal performing a plant function using stolen cellular machinery. The rhinophore structures on its head look like ears. They are chemical sensors used to detect food sources. A toxic, foul-tasting mucus deters predators. Its movement and shape mimic flatworms, providing secondary camouflage. Reproduction is hermaphroditic, ensuring population survival in isolated environments. Eggs are laid in perfect geometric spirals on algae surfaces. If a corrida is bitten off, it regrows and repopulates with new chloroplasts. Despite its solar technology, its brain is a simple cluster of ganglia. Its habitat spans the deep reefs of Japan, Indonesia, and the Philippines. 2 simple ocelli, eye spots, detect movement but cannot resolve complex images. The mainstream theory treats kleptoplasty as a metabolic supplement. The chloroplasts provide emergency energy during food scarcity. They are not a permanent transition to autotrophy. The comparative theory connects the sea sheep to the most important event in cellular history: endosymbiosis. Roughly 2 billion years ago, ancient cells incorporated free-living bacteria that became mitochondria. Later, other cells captured cyanobacteria that became chloroplasts. The sea sheep is doing the same thing right now. It suggests kingdom boundaries have always been permeable. The fringe interpretation calls it an organic drone, a self-sustaining solar-powered biological unit that defies traditional zoological categories. It raised the question of whether animal-plant mergers could be engineered, or whether they have been engineered before, by processes we do not yet understand. From an animal that steals sunlight, we move to one that survives where there is almost no oxygen at all. Number 8, the vampire squid, the oxygen problem. Vampiroteuthis infernalis. The vampire squid from hell. The name is misleading. It is not a vampire. It is not even technically a squid. It is the only living member of the order Vampiromorphida, phylogenetically suspended between octopuses and squids. And its defining feature is not the Gothic appearance, it is the address. The oxygen minimum zone. This is water where dissolved oxygen drops to as low as 3% saturation. Most complex animals cannot survive below about 40%. The vampire squid thrives at 3. Its blood is copper-based, blue, not red. Hemocyanin, the oxygen transport protein in its blood, is highly optimized to bind oxygen molecules in near-anoxic water. In modern chemical terms, copper-based hemocyanin has a lower oxygen affinity than iron-based hemoglobin, but functions more efficiently at low temperatures and low oxygen partial pressures. Its eye-to-body ratio is the largest of any animal on Earth. Eyes the size of a dog's and a body the size of a football. It is not a predator, it is a detritivore. 2 retractable filaments, packed with nerves, extend into the darkness to catch falling organic debris. Marine snow, dead plankton, fecal pellets, and decaying tissue drifting down from the surface. That is dinner. Body density is nearly identical to seawater. It hovers with 0 energy expenditure. Ammonium ions stored in its tissues maintain neutral buoyancy. When threatened, it pulls its arms over its head, revealing rows of fleshy cirri that look like spikes. The pineapple posture. It does not have a functional ink sac. Instead, it ejects bioluminescent mucus, a glitter cloud of light that distracts and confuses predators. Photophore tips on its arm ends pulse with light. It can dim or brighten them to mimic the size and distance of other animals. Visual hacking. 2 large ear-like mantle fins allow it to fly through the water like a deep-sea bird in slow motion. Its evolutionary timeline is staggering. 300 million years without significant morphological change. That predates the dinosaurs by roughly 70 million years. In the late Paleozoic, when atmospheric oxygen levels were fluctuating dramatically, this animal's ancestors were already perfected for low oxygen survival. A donut-shaped nerve ring brain surrounds its esophagus. Cellular membranes are highly fluid to prevent freezing or cracking under deep pressure. Mainstream theory calls it a metabolic minimalist. Every feature is an adaptation to survive where nothing else can. The fringe theory calls it a Gothic ancient, a survivor from a low-oxygen Earth whose body plan may represent what life looked like when atmospheric oxygen was a fraction of current levels, or what life might look like on low-oxygen exoplanets. If the vampire squid shows how little oxygen life actually needs, the next animal shows how little change life actually requires. Number 9, the frilled shark. 80 million years without an update. Clematicelicus anguineus carries 25 rows of trident-shaped teeth, approximately 300 needles arranged in backward-facing arcs designed to hook soft-bodied prey like squid. Its body is eel-like, long and cylindrical. It strikes forward like a snake, not sideways like most sharks. 6 pairs of frilled gill slits line its throat, edged with ruffled red tissue. This is likely the biological origin of multi-gilled sea serpent legends across maritime cultures. The species reaches about 6.6 feet, roughly 2 meters. Its skeleton is cartilaginous, not the dense cartilage of a great white. Low-density, flexible cartilage that compresses rather than cracks under abyssal pressure. This is not a modern shark. Its skeletal structure has remained largely unchanged for 80 million years. The gestation period is the longest of any vertebrate on Earth, 42 months, 3.5 years. A female frilled shark carrying young today conceived before the last United States presidential election cycle. The terminal jaw sits at the front of the head, not underslung like modern sharks. It can unhinge and swallow prey half its body size. Deep-sea suction feeding creates a vacuum that inhales soft-bodied animals whole. Its lateral line system runs the full length of its body, detecting movement in complete darkness through pressure wave sensitivity. A massive, oil-filled liver provides buoyancy. There is no nictitating membrane, the third eyelid found in modern sharks. In the abyss, there is no debris to protect against. It swims in an S pattern, undulating like a lungfish. Its thermal preference is cold water below 10 degrees Celsius. It can die of heat stress if brought to the surface. Low genetic diversity suggests long-term survival in a stable, unchanging niche across geological timescales. Mainstream theory explains this through evolutionary stasis. When an organism is perfectly adapted to a stable environment, selection pressure effectively stops. The niche has not changed, so neither has the shark. The speculative theory calls it the primordial monster, an entity that evolution forgot. Its deep-sea stability preserves a body plan that surface ecosystems would have eliminated through competition long ago. The question it raises for astrobiology is direct. Could similar stasis characterize life on geologically stable exoplanets or moons? If the frilled shark shows that life can stand still for 80 million years, the next organism shows that life can run backward. Number 10. The Immortal Jellyfish. Biological time reversal. Turritopsis dohrnii is 4.5 millimeters wide, smaller than a fingernail. It is the only known animal that can reverse its own life cycle. When stressed, starving, or physically damaged, it does not die. It reverts. Every specialized cell in its body, nerve cells, muscle cells, digestive cells, transforms back into a pluripotent stem cell. This process is called transdifferentiation. The adult medusa collapses into a cyst, attaches to a surface, and becomes a polyp, a juvenile. It then buds off multiple new clones and begins the cycle again. No other animal on Earth does this. Regeneration exists in many species. Salamanders regrow limbs, starfish regrow arms, but those are repairs. This is a complete reversal of the developmental program, a mature organism returning to an earlier life stage and starting over. The jellyfish begins with 8 tentacles as a juvenile and can develop up to 90 in its mature medusa form. A red digestive system is visible through the transparent bell. Stress triggers include temperature changes, food scarcity, and physical damage. During reversion, the metabolic rate and cellular processes slow to nearly zero. Unique gene expressions suppress senescence genes, the aging genes that exist in humans but are not silenced. An ongoing debate asks whether the new jellyfish retains any cellular memory from its previous iteration. The bell is highly elastic, allowing rapid, jerky propulsion. It operates on a nerve net rather than a brain. It has colonized oceans worldwide by hitchhiking in ballast water, functioning like a biological virus. Sticky tentacles cling to moving objects, facilitating intercontinental spread. The most resilient strains have been discovered in deep, cold basins. Barring predation or physical destruction, it has no biological end date, potentially immortal. Mainstream theory treats transdifferentiation as a stress response survival mechanism, a biological failsafe, not true immortality. Predation, disease, and environmental catastrophe still kill them routinely. The comparative theory suggests this capability may be a remnant of an ancient, more plastic cellular architecture. Early multicellular life may have been far more reversible. Teratopsis dohrna preserved what other lineages lost. The fringe theory frames it as the eternal hive, a species that cannot truly die, only reset, the ultimate biological insurance against planetary extinction events, and a model for what engineered biological immortality might eventually look like. 10 creatures. 10 sets of problems that physics says should be unsolvable. Now the question becomes how they solve those problems collectively, starting with the most basic: pressure. Pressure, darkness, and the physics of survival. Every 10 meters of ocean depth adds roughly 1 atmosphere of pressure. At 4,000 meters, organisms face 400 atmospheres. That is 400 times the air pressure at sea level. At those forces, proteins denature, they unfold, they stop functioning. The deep sea should be a dead zone. It is not, because multiple lineages independently evolve the same countermeasure. Piezolites. These are chemical stabilizers, small organic molecules like trimethylamine N-oxide that physically brace protein structures against compression. The Bigfin Squid uses them. The Glass Octopus uses them. They are convergent solutions to a universal physics problem. The Barreleye's fluid-filled dome is pressurized to match surrounding water, preventing structural collapse. The Vampire Squid's cellular membranes are unusually fluid, adapted to remain flexible rather than freezing or cracking. The Frilled Shark's low-density cartilage skeleton compresses rather than shatters. The Glass Octopus is entirely hydrostatic. Remove it from the pressure that shapes it, and the body disintegrates. Buoyancy strategies vary: gas bladders in the Barreleye, oil-filled livers in the Frilled Shark, ammonium ions in the Vampire Squid. Traditional swim bladders, the standard solution for surface fish, are unreliable at extreme depth because gas compresses. Most deep-sea species abandon them entirely. Below 1,000 meters, light disappears completely. The sensory world shifts from vision to mechanoreception, chemoreception, and bioluminescence detection. The Frilled Shark's lateral line system and the Bigfin Squid's mechanoreceptors function as pressure wave detectors, replacing eyesight with vibration mapping. Counter-illumination, producing ventral light to cancel one's own shadow, is a shared strategy across multiple species. Slow metabolism unifies survival across the Barreleye, the Vampire Squid, and the Bigfin Squid. Low oxygen, low temperature, and low food availability all demand radical energy conservation. The deep sea is the largest habitat on Earth by volume. It remains less explored than the surface of the moon. Mainstream theory sees these adaptations as convergent solutions to universal physical constraints. The comparative theory notes that deep sea stability may preserve body plans from the Precambrian or Paleozoic era, designs no longer viable in surface ecosystems. The fringe theory observes that the physical conditions of the deep ocean, high pressure, low light, low oxygen, more closely resemble predicted conditions on icy moons like Europa than they resemble Earth's surface. Pressure is one shared problem, light is another, and in the deep sea, light is not just vision, it is language. Bioluminescence, the language of the abyss. An estimated 76% of deep sea organisms produce their own light. Bioluminescence is the rule, not the exception. The chemistry is elegant. A substrate called luciferin reacts with an enzyme called luciferase. The reaction produces photons with almost zero heat. Cold light. The Barreleye's green eye filters are specifically tuned to isolate bioluminescent wavelengths from ambient light. The Vampire Squid's photophore control system dims and brightens to mimic the apparent sizes and distances of other organisms. The Siphonophore's neon sign zooids are specialized colony members whose sole function is generating light to attract prey. The Vampire Squid replaces ink with a cloud of bioluminescent glitter, mucus. A confusion mechanism made of light instead of darkness. Blue and green wavelengths dominate deep-sea bioluminescence because they travel farthest in seawater. Red bioluminescence is rare, and that rarity is significant. Most deep-sea organisms cannot see red light. Species that produce it operate on a private frequency. An invisible channel, a biological encrypted communication line. The Bigfin Squid takes a different approach entirely, no photophores, no pigment-based camouflage. It relies on stillness and near-total transparency. The sea sheep's chlorophyll-based green glow looks similar to bioluminescence, but operates through a completely different mechanism. Solar energy storage, not chemical reaction. The Bobbit Worm detects light changes across its entire body without producing any light itself. The Glass Octopus neither produces light nor reflects it. It solves the bioluminescence problem by not participating. The Frilled Shark absorbs light. Its dark coloring, chocolate brown to charcoal gray, is passive light management. The Immortal Jellyfish's transparent bell is a non-bioluminescent stealth strategy similar to the Glass Octopus. There is an arms race at work. Prey evolve to detect bioluminescent lures. Predators evolve more sophisticated light patterns. Human ROV operations now introduce light pollution into these environments, disrupting natural bioluminescent signaling with consequences we are only beginning to study. Mainstream theory holds that bioluminescence evolved independently at least 50 times across the tree of life. It is the most convergent complex trait known to science. The comparative theory notes that the chemical precursors for bioluminescence, various luciferin variants, appear to predate complex animals. Light production may be an ancient metabolic byproduct that was co-opted for communication over hundreds of millions of years. The fringe theory imagines the deep ocean hosting a light-based internet, a signaling network so vast and interconnected that we are only beginning to detect its edges, and disrupting it may carry ecological consequences we cannot predict. If bioluminescence is the deep sea's language, then DNA is its filing system, and some of these creatures are filed in categories that do not exist. Taxonomy, DNA, and creatures that defy classification. The Vampire Squid is the only living member of the order Vampiromorphida. It sits between octopuses and squids on the phylogenetic tree, belonging fully to neither. The Barreleye is the sole recognized species in its genus. Macropinna has no close relatives. The Frilled Shark's low genetic diversity points to survival in a stable niche with minimal population mixing across geological timescales. The Siphonophore presents a classification problem that is not genetic but philosophical. Is a colony of genetically identical, specialized zooids one organism or many? Taxonomy was not built for this question. The Mimic Octopus's RNA editing recodes its own RNA without DNA mutation. That is adaptation that challenges the gene-centric view of evolution. The organism changes its protein function without changing its genome. The Bobbit Worm shows extreme genetic divergence from other polychaete worms, suggesting a lineage so ancient and so isolated that standard classification groups strain to contain it. The sea sheep's kleptoplasty is a taxonomic boundary violation. An animal performing a plant function using stolen cellular machinery. Cephalopod. RNA editing produces phenotypic flexibility without genetic change, potentially decoupling evolution from DNA mutation in ways that challenge the modern synthesis. The Frilled Shark's cartilaginous skeletal structure has remained primitive for 80 million years. The Bigfin Squid family Magnapinnidae was not even recognized until the late 1990s. Classification sometimes lags discovery by decades. The Glass Octopus is an evolutionary departure. It abandoned the entire cephalopod defensive toolkit, ink, chromatophores, shape-shifting, for a single strategy. The Siphonophore's evolutionary lineage is one of the oldest for complex life, remaining largely unchanged. Does extreme specialization represent evolutionary success or a dead end? Mainstream theory treats taxonomic outliers as the far ends of natural variation, products of long isolation and niche stability. The comparative theory observes that many of these lineages diverge during periods of mass extinction, occupying niches vacated by dominant groups. They are survivors, not oddities. The fringe theory points to the 2018 paper in Progress in Biophysics and Molecular Biology that applied the panspermia hypothesis to cephalopods, suggesting octopus eggs or genetic precursors could have arrived via cometary material. Whether or not one takes that seriously, The convergence of so many classification-defying traits in the deep sea demands an explanation. Our taxonomic system was built for surface life. The deep sea may require an expanded framework, and if our classification system struggles with Earth's own ocean, it raises an immediate question about how we would classify life found somewhere else entirely. What the deep sea tells us about elsewhere. If life exists on Europa or Enceladus, it likely inhabits subsurface oceans under ice. Those environments are closer to Earth's deep sea than to its surface. The vampire squid's survival at 3% oxygen saturation demonstrates that complex life can operate in near anoxic conditions, directly relevant to low oxygen extraterrestrial environments. Europa's ocean may experience pressures comparable to Earth's hadopelagic zone, where the Bigfin Squid has been filmed at over 6,000 meters. The Siphonophore's modular colonial architecture offers a model for how complex life might organize without a central nervous system. Non-brain-based intelligence. The Immortal Jellyfish's transdifferentiation offers a survival strategy for organisms facing extreme environmental variability, a plausible adaptation for worlds with erratic conditions. Bioluminescence proves that complex ecosystems can operate entirely without sunlight. The deep ocean runs on chemosynthetic and bioluminescent energy. The sea sheep's kleptoplasty demonstrates that kingdom boundaries are not fixed. Extraterrestrial life need not be strictly animal or plant. The glass octopus's transparency solves a predation problem without pigment. That strategy could evolve in any light-poor liquid environment. The frilled shark's 80 million year stasis shows that stable environments produce stable organisms. Geologically quiet moons might harbor life that has not changed in timescales that dwarf anything on Earth's surface. The Bobbit Worm's purely mechanosensory and chemosensory hunting is a model for predation without vision, directly applicable to lightless subsurface oceans. NASA's interest in deep-sea analogs for Europa mission design has increased significantly in recent years. Hydrothermal vent ecosystems are the closest Earth analog to hypothesized subsurface ocean energy sources on icy moons. The chemical conditions around deep-sea vents, hydrogen sulfide, extreme temperatures, heavy metals, overlap with predicted conditions on Enceladus. The Mimic Octopus's decentralized intelligence models distributed cognition, relevant to speculation about non-centralized alien nervous systems. The Barreleye's green filter eyes demonstrate that sensory systems can be precisely tuned to environmental light conditions, however exotic. Mainstream theory holds that deep-sea organisms are powerful analogs for astrobiological modeling, but analogy is not homology. Similar environments produce similar solutions through convergent evolution, not shared origin. The comparative theory frames Earth's deep sea as a time capsule. Low oxygen, low light, high pressure. These conditions characterized earlier epochs on Earth. Studying the deep sea is effectively studying the past, and that past may resemble other planets' present. The speculative theory offers the strongest version. Not that these creatures are aliens, but that the deep ocean's independence from solar energy makes it functionally an alien biosphere operating under alien rules within our own planet. The practical value of that framing is not belief in extraterrestrial origin. It is expansion of the search parameters for what life can look like. The deep sea is simultaneously the most alien and most terrestrial environment we know. Innovation at the bottom of the world. Every creature in this series solves an impossible physics problem. Pressure, darkness, oxygen deprivation, food scarcity. The solutions are radically diverse: transparency, immortality, colonial architecture, photosynthesis theft, bioluminescent deception, shape-shifting intelligence, 80 million year stasis. None of these were designed. They emerge from constraint. The deepest ocean is also the least explored habitat on Earth, less mapped than Mars. Each decade of ROV technology reveals body plans and survival strategies previously unknown to science. The Bigfin Squid was not recognized until the 1990s. The Barreleye's dome was not documented until 2004. Discoveries continue. The alien label applied to these creatures reflects a failure of imagination, not a failure of taxonomy. The deep sea demonstrates a principle that extends far beyond biology. The harshest environments produce the most creative solutions. That principle is observable in engineering, in architecture, in technology. Extreme constraint drives radical innovation. If we want to understand what life could look like elsewhere in the universe, We should look down before we look up. The fundamental laws of physics are the same everywhere, and life, wherever it exists, must negotiate with them. These 10 creatures are not evidence of extraterrestrial origin. They are evidence of what terrestrial life is capable of when pushed to its absolute limits. The ocean's deepest zones remain the planet's greatest repository of undiscovered biology. Understanding these organisms has direct applications in materials science, in medicine through transdifferentiation research, and in robotics through decentralized intelligence models. The question that runs through everything we have covered is simple. If we found these creatures on Europa, would we call them alien? We already call them alien here. The word alien means belonging to another. And the deep sea is, in every practical sense, another world, 300 million years old, drifting in 3% oxygen, indifferent to whatever watches from above.